In this video, we'll be looking at enzyme structure and enzyme activity. Enzymes are proteins that are biological catalysts. Enzymes increase the rate of reactions by lowering the activation energy. For any reaction to take place, the reactants must collide with energy equal to or greater than the activation energy. By lowering the activation energy, enzymes make it more likely for a successful reaction to take place. As enzymes are made of proteins, they have a primary structure, which is the sequence of amino acids. They also have a secondary structure, which leads to a very specific tertiary structure. Enzymes have an active site, which is where the substrate fits. The active site has a specific shape and is usually complementary to the substrate. Substrate is fit into the active site by the induced fit method. When the substrate comes into close proximity with the active site, the charges present on the substrate slightly alter the shape of the active site. This allows the substrate to fit into the active site. This is known as the induced fit model because the substrate induces a change in the shape of the active site. This change in shape is temporary. When a substrate enters the active site of the enzyme, an enzyme-substrate complex is formed. It is necessary for an enzyme-substrate complex to be formed for a reaction to take place. The active site of the enzyme is complementary to the substrate. The specific shape of the active site is caused by the folding and bonding of the tertiary structure of the protein. The amino acids in the active site are held in place by van der Waals forces, hydrogen bonds, ionic bonds, and disulfide bridges. If any of these bonds or interactions are broken, it will alter the tertiary structure of the active site, which will damage the shape of the active site. pH is one factor that affects the rate of enzyme activity. As you can see in the graph, each enzyme has an optimum pH, which is where the rate of reaction is fastest. Away from the optimum pH, the rate of reaction begins to decrease. pH is a measure of hydrogen ion concentration. When the pH is acidic, there are more hydrogen ions. When the pH is more alkaline, there are more hydroxide ions. Hydrogen and hydroxide ions both have a charge. Hydrogen ions are positively charged. Hydroxide ions are negatively charged. When these charged particles come into close proximity with the active site, they will affect the hydrogen bonds, van der Waals forces, and ionic bonds in the active site. This will lead to a change in the tertiary structure of the enzyme and the active site being altered. No enzyme-substrate complexes can be formed, so the rate of reaction decreases. Temperature also affects the rate of reaction. Each enzyme has an optimum temperature, which for humans is 37 degrees. When the temperature is first increased, the kinetic energy of the enzymes and substrate increases. This means more enzyme-substrate complexes are formed, leading to a faster rate of reaction. Increasing the temperature even more will break the hydrogen bonds and van der Waals interactions in the active site. This alters the active site and changes the tertiary structure of the enzyme, so less enzyme-substrate complexes are formed, reducing the rate of reaction. When increasing enzyme concentration, the rate of reaction will increase as more enzyme-substrate complexes are formed. The rate will continue to increase until the substrate concentration becomes the limiting factor, meaning there are more enzyme active sites available than the number of substrate molecules. Increasing the substrate concentration also increases the rate of reaction. This is because there are more substrate molecules available, so more enzyme-substrate complexes are formed, leading to a higher rate of reaction. The rate of reaction continues to increase until the enzyme active sites become a limiting factor. The key points in this video are: enzymes and substrates join together to form enzyme-substrate complexes. Enzymes have an active site which has a specific tertiary structure and is complementary to the substrate. The substrate fits into the active site by the induced fit model. Enzymes can become denatured by extremes of pH or temperature. This causes a change in the tertiary structure of the enzyme, which alters the shape of the active site, meaning less enzyme-substrate complexes are formed. An exam tip is, when a rate of reaction graph is shown and you're asked to calculate the rate, you need to draw a tangent to the graph.